Hello everyone, and welcome. I'm excited to kick off this session on the chemistry of nutrients. Today, we'll be focusing on Module 2, specifically the chemistry of amino acids and proteins. This is part of our BCHM 241 course on the biochemistry of macromolecules. Over the next few minutes, we'll explore the fundamental aspects of amino acids and proteins, including their structure, properties, and roles in biology. Let's get started and dive into this fascinating topic. Let's start by disrupting the tissue or cells to release the proteins we want. We can do this mechanically using a homogenizer, which physically breaks apart the tissue. Alternatively, we can freeze and thaw the tissue repeatedly, which also helps to break open the cells. Chemicals like saponin and SDS are also used. They help to solubilize the cell membranes and release the proteins. Once we've done that, the next step is to get a homogeneous solution. To do this, we need to remove the cell debris and any insoluble material. We can do this by filtering the mixture through muslin cloth or filter paper, which helps to separate out the larger debris from the soluble proteins. At this point, what we have is called a crude extract. It contains the protein or enzyme of interest, but it's mixed with other proteins and cellular components. So, this is just the beginning. Further purification is needed to isolate our target protein. This step is crucial because it sets the foundation for successful protein isolation and ensures we work with a cleaner sample moving forward. Today, I want to introduce you to the concept of differential centrifugation. This technique is fundamental in cell biology and biochemistry for separating cellular components. The key idea is that different cell parts have different sizes and densities, so they sediment at different speeds when we spin them at various centrifugal forces. By carefully adjusting the speed and duration of centrifugation, we can selectively isolate specific organelles or structures. This method is powerful because it allows us to break down complex mixtures into more manageable, purified components. It's a cornerstone technique that helps us understand the structure and function of cells in much greater detail, tying into our broader goal of exploring cellular organization and function. Density gradient separation is a fascinating technique that allows us to separate components based on their density differences. Essentially, we create a gradient, usually with a substance like sucrose or cesium chloride, where the density gradually changes from one end to the other. When we spin our sample through this gradient in a centrifuge, each particle moves to the point where its density matches that of the surrounding medium. This method is particularly useful because it provides a highly effective way to isolate specific cell types, organelles, or even molecules with high precision. It's a key tool in many biological and biochemical applications, helping us to analyze complex mixtures and understand the composition of biological samples in greater detail. Connecting this to our overall theme, density gradient separation exemplifies how physical principles can be harnessed to advance our ability to study and manipulate biological systems more effectively. Proteins are soluble in aqueous media because they have hydrophilic amino acid side chains facing outwards, which can interact with water. These hydrophilic side chains are provided by specific amino acids. For example, the basic amino acids like arginine, histidine, and lysine contribute to this, as do the acidic amino acids such as aspartate and glutamate. Additionally, neutral hydrophilic amino acids like asparagine, glutamine, serine, threonine, tyrosine, and cysteine also play a role. Now, if any compound interferes with these interactions, say, by reducing the amount of available water, the solubility of the protein will decrease. Essentially, less interaction with water means the protein is less likely to stay dissolved. As these interactions lessen, protein-protein interactions become more prominent, leading to aggregation and eventual precipitation out of solution. However, as long as we keep the temperature low, around 4 degrees Celsius, we can prevent irreversible denaturation of the protein. This means that the precipitated proteins can often be redissolved in buffer later on, which is useful for purification purposes. When we talk about ammonium sulfate precipitation, it's a classic method that takes advantage of this principle. By adding ammonium sulfate, we reduce the protein's solubility selectively, causing it to precipitate out, which we can then collect and redissolve as needed, provided the conditions are carefully controlled. Let's talk about dialysis and why it's such an important step in protein purification. Dialysis is commonly used to remove salts, like ammonium sulfate, from proteins. These salts can interfere with downstream processes or analyses, so getting rid of them is crucial. Now, the key idea here is that dialysis involves the passage of solutes through a semi-permeable membrane. Think of this membrane as a filter that allows smaller molecules to pass through, while larger ones, like the proteins themselves, stay behind. The pores in the dialysis membrane are of a specific size, which determines what can and can't pass. Water, salts, protein fragments, and other small molecules, those smaller than the pore size, can pass through easily. But the proteins, being larger, remain inside the dialysis bag. This process effectively separates the proteins from unwanted small molecules, like salts, allowing us to purify our proteins further. Understanding how dialysis works helps us appreciate its role in maintaining protein integrity while removing impurities, which is central to our overall goal of obtaining high-quality proteins for our research or applications. Now, let's talk about how separation using column chromatography can be based on different properties of the molecules we're trying to isolate. First, size plays a crucial role. This is where gel filtration or size exclusion chromatography comes in. It separates molecules based on their size, with larger molecules eluting first because they don't enter the pores of the gel, while smaller ones take longer as they pass through these pores. Next, charge is another key factor. Cation exchange chromatography separates proteins based on their charge properties. Proteins with different charges will bind to the charged matrix with varying affinities, allowing us to elute them separately by changing the salt concentration or pH. Then there's affinity chromatography, which is perhaps the most specific method. It relies on the natural affinity between a protein and a particular ligand attached to the chromatography matrix. This allows us to isolate a specific protein from a mixture with high selectivity. As a result of these separation techniques, we usually obtain several fractions, each containing different proteins. This step is essential in purification processes, helping us progressively enrich our target protein. And that's the essence of chromatography and purification, using different properties like size, charge, or affinity to effectively separate and isolate proteins, moving us closer to obtaining a pure sample for further analysis. Now, let's talk about purification and electrophoresis. These are crucial steps in analyzing and isolating specific molecules, especially proteins and nucleic acids. Purification involves separating the desired molecule from a complex mixture to obtain a pure sample, which is essential for accurate downstream analysis. Electrophoresis, on the other hand, is a technique that uses an electric field to move charged molecules through a gel matrix. This allows us to separate molecules based on their size and charge. It's a powerful method because it gives us a visual representation of the different components, making it easier to identify and analyze the molecules of interest. Together, these techniques are vital tools in molecular biology, helping us to refine our samples and better understand their properties. They connect directly to the overall theme of understanding biological molecules in greater detail. Now, let's talk about how we determine the primary structure of a polypeptide, which essentially means identifying and quantifying its amino acids. The process starts with hydrolysis, where we break down the protein into its individual amino acids. We do this by treating the protein with a strong acid, specifically 6N-HCl, and heating it at around 110 degrees Celsius for 24 to 72 hours. This high-temperature acid hydrolysis ensures the protein is fully broken apart. Once hydrolyzed, the next step is separation. We use cation exchange chromatography to separate the individual amino acids. This technique takes advantage of the charge differences among amino acids, allowing us to isolate each one effectively. After separation, detection becomes crucial. We typically detect amino acids through a reaction with ninhydrin, which produces a color change, either purple or blue, absorbing light at 570 nanometers, or a yellow color absorbing at 440 nanometers. This colorimetric detection helps us quantify the amino acids present. To facilitate this process, we often use an amino acid analyzer. These instruments incorporate ion exchange chromatography or high-performance liquid chromatography, or HPLC, combined with sensitive detection systems. This setup allows us to analyze each amino acid precisely and efficiently. Finally, understanding the amino acid composition of a protein gives us valuable insights into its structure and function. By knowing which amino acids are present and in what amounts, we can better understand the protein's properties and how it contributes to biological processes. Let's talk about Sanger sequencing. In 1953, Frederick Sanger made a breakthrough by determining the sequence of the hormone insulin. This was a landmark achievement in the field of molecular biology. Sanger used a specific reagent called 1-fluoro-2,4-dinitrobenzene, which reacts with the amino group at the amino terminus of the peptide. This reaction allows us to identify the terminal amino acid of the peptide chain. It's interesting to note that other reagents, such as dansyl or dabsyl chloride, can also be used for similar purposes. These reagents help in labeling the terminal amino acids, making sequencing possible. Once the peptide is labeled, it can be hydrolyzed to release the derivative. By analyzing this derivative, we can determine the identity of the terminal amino acid. However, it's important to remember that this method primarily identifies only the terminal amino acid and the number of amino acids in the peptide chain. It doesn't give us the full sequence of the entire protein, but it is still a crucial step in understanding protein structure. This method laid the foundation for peptide sequencing and helped us understand the building blocks of proteins, which is essential for many areas of research and medicine. Today, I'll be talking about Edmonds degradation, which is a classic method for sequencing amino acids in a peptide. The key reagent used here is phenylisthiocyanate, commonly known as Edmonds reagent. Now, what makes this method quite elegant is that it labels the amino terminal residue of the peptide and then cleaves it off without disturbing the rest of the peptide bonds. This means we can isolate the N-terminal amino acid, identify it, and then repeat the process to determine the next residue. This cycle continues until we've identified all the amino acids in the sequence. However, it's important to note that this method is most effective for relatively short sequences of amino acids. It's not suitable for very long peptides. Another interesting point is that this process can be automated using a machine called a sequenator, which greatly speeds up the sequencing process and improves accuracy. So, in summary, Edmonds degradation is a powerful tool for peptide sequencing, especially for short sequences, and it relies on a chemical reaction that allows us to sequentially identify amino acids from the N-terminus. This technique is integral to understanding protein structure at the amino acid level and connects directly to our overall theme of protein analysis. This slide doesn't contain enough information. That's why we're moving to the next slide. Let's talk about breading disulfide bridges, which are important in stabilizing protein structures. When we're sequencing large peptides, one of the key steps involves ensuring these disulfide bonds are correctly identified and preserved. This process helps us understand the peptide's three-dimensional conformation, which is crucial for its biological activity. Now, in the context of sequencing, handling disulfide bridges can be tricky because they can link different parts of the peptide chain, making the sequence harder to interpret. So, we often need to carefully reduce these bonds to break them apart, analyze the fragments, and then sometimes reoxidize them to confirm the connections. This step is vital because disulfide bonds can mask certain amino acids or regions, leading to incomplete or incorrect sequencing if not properly managed. By systematically breaking and then rebuilding these bridges, we ensure a more accurate and complete peptide sequence. In the broader scope of peptide sequencing, mastering the handling of disulfide bridges helps us piece together the structure more precisely, which ultimately supports our understanding of how the peptide functions biologically. Many polypeptides are quite large, often composed of more than 100 amino acids. Because of their size, it can be challenging to analyze their structure directly. So one common approach is to cleave these molecules at specific sites. By doing this, we break them down into smaller, more manageable fragments. To do this effectively, we often use more than one cleaving agent, such as specific enzymes or chemicals, each targeting different sites. This method allows us to sequence the resulting fragments more easily. Essentially, by breaking the polypeptide into smaller pieces, we can better understand its overall structure and amino acid sequence, which ties into our broader goal of characterizing protein molecules accurately. Now, let's talk about how Edman degradation can be used to determine the amino acid sequence of each fragment. This technique is quite powerful because it allows us to identify the sequence step by step, starting from the N-terminus of the peptide. Essentially, Edman degradation selectively removes the first amino acid, which we can then identify, and then the process repeats, peeling away one amino acid at a time. This way, we can piece together the entire sequence of the peptide fragment. It's a crucial method in protein sequencing, especially when working with smaller peptides. By understanding the amino acid sequence, we gain valuable insights into the protein's structure and function, which ties back to our overall theme of understanding protein composition and behavior. Now, when it comes to cleaving agents, it's important to recognize that more than one can be used in the same experiment. For example, trypsin and pepsin are commonly employed because they cut at different sites, giving us more information about the protein. The fragments we obtain from these cleavages can then be compared after sequencing. By analyzing the sequences of these fragments, we can identify regions that overlap. These overlapping sequences are key because they help us determine how the fragments fit together. Essentially, by tracing these overlaps, we can reconstruct the original sequence in the correct order. This process of ordering the overlapping sequences is crucial in protein sequencing, as it allows us to piece together the puzzle and understand the full structure of the protein. So, using multiple cleaving agents and analyzing overlapping sequences provides a more complete and accurate picture of the protein's structure. Let's take a closer look at the structural organization of proteins. Proteins are incredibly complex molecules, and their function is closely tied to their structure. The basic building block of a protein is the amino acid, which links together in specific sequences to form a polypeptide chain. This chain then folds into a unique three-dimensional shape, which is crucial for its activity. There are several levels of organization to keep in mind. First, the primary structure is simply the sequence of amino acids. This sequence determines how the protein will fold and what shape it will take. Next, the secondary structure refers to local folded regions, like alpha helices and beta sheets, stabilized mainly by hydrogen bonds. Moving on, the tertiary structure describes the overall three-dimensional shape of a single polypeptide chain, including how the secondary structures come together. This folding is driven by various interactions like hydrophobic effects, ionic bonds, and disulfide bridges. Finally, some proteins have a quaternary structure, where multiple polypeptide chains come together to form a functional protein complex. Understanding this organization helps us grasp how proteins perform their diverse functions in the body, from catalyzing reactions to providing structural support. It's this intricate folding and organization that makes proteins such a fascinating and vital part of biological systems. Let's take a closer look at secondary structure. Essentially, this refers to the local folding patterns within a protein, which are stabilized by hydrogen bonds. The two most common types are alpha helices and beta sheets. These structures are fundamental because they give the protein its shape and influence its function. Understanding secondary structure helps us see how a protein's shape relates to its role in the body. Recognizing these patterns is key to grasping the bigger picture of protein structure and how it all fits into the overall theme of biological function. Now, let's talk about the tertiary structure of a protein. This is essentially the total three-dimensional shape of the entire polypeptide chain. It includes alpha helices, beta sheets, and any loops, turns, or bends that help give the protein its specific shape. The key point here is that this overall conformation depends heavily on the properties of the R groups, or side chains, of the amino acids. These properties influence how the chain folds and interacts internally. The folding process can happen spontaneously, meaning the protein naturally assumes its correct shape, or with the assistance of molecular chaperones, which help guide the folding process when needed. Once folded, the structure is stabilized by various types of bonds, covalent bonds like disulfide bridges, and non-covalent interactions such as hydrogen bonds and van der Waals forces. These interactions work together to maintain the protein's shape. An interesting aspect to note is how the protein's environment influences its structure. Hydrophobic side chains tend to bury themselves inside the molecule, away from water, while hydrophilic groups are found on the surface, interacting with the aqueous environment. This arrangement is crucial for the protein's stability and function. Overall, the tertiary structure is vital because it determines how the protein functions in the cell. It's a complex but beautifully coordinated process that relies on both the chemical properties of amino acids and the cellular environment. Now, let's talk about the quaternary structure of proteins. This is all about how multiple folded protein or coiled protein molecules come together to form a multi-subunit complex. Essentially, when a protein has several polypeptide chains, it gains an extra level of structural organization, what we call the quaternary structure. This level describes the spatial relationship between these individual subunits within the whole protein. Think of it as how the pieces fit together in a complex puzzle. Understanding this arrangement is crucial because it often influences the protein's function and stability. So, in summary, the quaternary structure is all about how these multiple chains interact and organize in a three-dimensional space, which is key to understanding how proteins work in the biological systems we're studying today. This slide doesn't contain enough information. That's why we're moving to the next slide. Now, let's take a closer look at the diverse roles that proteins play in biological systems. First, we have structural support, proteins like collagen, which provides strength and structure to our skin and bones, and keratin, which is essential for the strength of our hair and nails. These proteins are fundamental to maintaining our body's shape and resilience. Moving on to catalysis, proteins such as amylase are crucial for digestion, helping to break down starches into sugars. DNA polymerase is another key enzyme, vital for DNA replication, ensuring genetic information is accurately copied during cell division. These enzymes speed up chemical reactions that are essential for life. Next, we have transport and storage functions. Hemoglobin, for example, is responsible for transporting oxygen from our lungs to tissues throughout the body, while ferritin stores iron, making it available when the body needs it. These proteins are essential for maintaining proper levels of vital nutrients and gases. In signaling and communication, insulin is a well-known hormone that regulates blood sugar levels by signaling cells to take up glucose. Receptor tyrosine kinases are involved in cell signaling pathways, helping cells respond to various stimuli and coordinate activities. These proteins enable cells to communicate effectively and respond appropriately to their environment. When it comes to defense and immunity, antibodies are proteins that specifically recognize and neutralize pathogens like bacteria and viruses. Complement proteins work alongside antibodies to destroy microbes directly, forming a crucial part of our immune defense system. Finally, in movement, actin and myosin are the proteins responsible for muscle contraction, enabling movement and force generation. Tubulin forms the microtubules that facilitate various types of cell movement and shape changes. All these examples highlight how versatile and vital proteins are. They're involved in almost every aspect of biological function. Understanding these roles helps us appreciate the complexity and elegance of life at the molecular level. Now, let's explore 3 common analytical methods used for detecting and quantifying proteins in a sample. First up, we have spectrophotometry, which is based on measuring the absorbance of light by the protein, usually at 280 nanometers. It's a quick and straightforward method, but it requires that the sample is relatively pure because other substances can also absorb at this wavelength. Next, there's the Bradford assay, a colorimetric technique that involves a dye binding to proteins and producing a color change, which we can then measure spectrophotometrically. This method is popular because it's fast, sensitive, and relatively easy to perform, making it suitable for many lab settings. Finally, we have enzyme-linked immunosorbent assay, or ELISA. This is a highly specific method that uses antibodies to detect and quantify a particular protein within a complex mixture. It's especially useful when you need to identify a specific protein among many others, such as in diagnostic tests or research. Connecting all these methods to our overall theme, each has its strengths and limitations, and selecting the right one depends on factors like sample purity, required sensitivity, and specificity. Understanding these techniques allows us to choose the best approach for accurate protein analysis in diverse applications.